Dear students, okay, so cell cycle we were discussing. Today we will discuss about the mitosis, and then in the next lecture we will discuss about the meiosis. Let's see what is mitosis. Before going for the mitosis, let's see a question. Centromere splits during a different phase is given over here. So what are the phases they have given? Prophase, metaphase, anaphase, telophase, and interphase. Right. So let's go with the interphase first. What happens during the interphase? Interphase we further divide into how many phases? G1, S, G2, and M phase. Yes, and in between the G1 there may be a G0 phase, and then it enters into the G1 again. Right. So what happens during the interphase? The interphase has the S phase. During the S phase, there is DNA synthesis occurs. DNA synthesis occurs. So during this DNA synthesis, all the DNA gets replicated. All the DNA gets replicated. And what is this one? The centromere. Centromere is the central non-coding DNA sequences, right? So this is our centromere. So see, during the S phase, the DNA replication occurs. That means the centromere replication occurs during which phase? S phase. S phase, the centromere also replicates. Two centromere becomes, but these two centromeres, they do not get separate during the S phase. They remain attached in the middle with a protein that is called cohesin. Right? So when the interphase completes, the cell enters into the M phase. And in the M phase has multiple 4 stages. What are those? P, M, A, T. What are those? P for prophase, metaphase, anaphase, and telophase. Prophase is the longest phase, right? So it undergoes the preparation for the mitosis or the meiosis. Then metaphase, during the metaphase, all the chromosomes, they arrange in the equatorial plane like this, right? So when we reach to the anaphase, these sister chromatids, they will move apart on two sides. So when they begins to move apart, this centromere has to get separated from the cohesin protein, so that half of this chromosome, this will move to one pole, the half of this will move to another pole. That means the arrangement occurs during the M phase, but when it reaches to the A phase means what? This is what? Prophase. M phase, I mean here it's the metaphase. A for what? Anaphase. So during the M phase, this chromosome, they remain arranged in the equatorial plane. When they enters into the anaphase, they begins to move to the opposite side. That means this centromere, which was already replicated, it gets separate during which phase? Anaphase stage. So after that, what happens? The telophase. During the telophase, again the nuclear membranes, they begin to reappears, right? So that the two nucleus will appear in the two poles of the cells, right? So in these two poles of the cells, we have two nucleoli appears during the telophase, right? So that the centromere splits into the two during which phase? Anaphase stage. During the metaphase, what happens? Equatorial arrangement occurs. During the prophase, what happens? It undergoes the preparation for the M phase. That means for the mitotic or meiotic divisions. During the telophase, what happens? Two daughter nucleoli appears, two daughter nucleoli. And during the interphase, it undergoes what? G1, S, G2, and M phases. So that hope you understand the answer will be what? The anaphase. Let's go to the introduction to the cell. What is cell? Cell is the functional plus structural unit of the life, right? Living organisms. This functional and structural unit, they will make what? Tissue will make the organ will make the organ system will finally make the whole organism, organism. Hope you understand. Yes or no? How many cells do we have in our body? We have about 40 trillion cells. 40 trillion cells. But these 40 trillion cells are of different, more than 100 physiological types, right? So those all 40 trillion cells that derive from the single zygote. So the question is here, even though they are derived from the same single zygote, they must be of same function and same structure, but why do we have more than 100 varieties? Because these cells, when they begin to divide, the zygote begin to divide and forms the multiple cells, these cells undergoes differentiation. Differentiation means these cells, they begin to adopt the different function and they begin to adopt the different structure, so that they become structurally and functionally different wherever they come, according to the tissues where do they present, right? So this is about the introduction to cell. And look at here, the cell composed of what? Cell membrane. And cell membrane is what? It's a phospholipid. It's a phospholipid bilayer, right? Phospholipid bilayer, the inner leaflet and the outer leaflet. You can see here two layers of the phospholipid. One phospholipid layer is this one, another phospholipid layer is this one, right? So we have two layers of the phospholipid. You can see the head inside, tail here. You can see the head outside and the tail inside here. Yes, so you can see this is E phase. We can divide this as E phase of the cell membrane. This is P phase of the cell membrane. So towards the P phase we have one layer of the phospholipid molecules. Towards the E phase of the cell membrane we have the another layer of the phospholipid molecules, right? And the phospholipid molecules has what? Head and tail, right? Head and tail. This tail are made up of what? Saturated and unsaturated fatty acids. So this is the cell membrane, and it also consists of what? Cell organelles. Cell organelles, we divide them into two groups, non-membranous and membranous. Non-membranous you can remember as CRPN, right? So these non-membranous cell organelles are what? CRPN. C for centrioles, or maybe cilia, right? Similarly, R you can remember as what? The ribosomes. R for what? Ribosomes. And you have P for proteasomes. And you have one N for nucleolus. Also you have the flagella. So all of these are what? Non-membranous cell organelles. So question in the exam. The rest of the others, all rest are what? Membranous cell organelles. So hope you understand. Yes, so the cell composed of what? Cell membrane and the cell organelle. Cell membrane consists of phospholipid bilayer. This phospholipid bilayer means two layers, right? So this one layer forms the E phase, one layer forms the P phase of the cell membrane. What is that E phase and the P phase of the cell membrane? See, this is my cell, this is inside of the cell, this is outside of the cell. The phospholipid bilayer which is present outside of the cell, this is what? E phase. And the phospholipid bilayer which is present inside of the cell, that is what? P phase. P phase means protoplasmic phase. E phase means extracellular phase. It is outside. Hope you understand. And the cell organelles we divide into two groups, that is what? Non-membranous and membranous cell organelles. And if you can remember non-membranous as CRPN, C for centrioles or maybe cilia, and R for ribosomes, P for you have the proteasomes, N for nucleolus, and maybe also flagella, right? All of these are what? Non-membranous cell organelles. Except these, rest of the other cell organelles are membranous cell organelles. So you can remember this. This is the cell. You can see the structure of the cell. Nucleus, you can see nucleolus over here, and you can see the, what is this one? Endoplasmic reticulums, which has the lots of ribosomes with them. So they are what? Rough endoplasmic reticulums. Outside of this you can see smooth endoplasmic reticulums which do not have the ribosomes on them. You can see the Golgi apparatus, the vesicular, the stack-like cisterns present. And let's go to the cell cycle now. What is cell cycle? Cell cycle is it follows the sequence of events. events which leads to the formation of what? Formation of daughter cells. Daughter cells. That's what we call as the cell cycle, right? So the cell cycle, finally the result of the cell cycle is what? Formation of daughter cells. These daughter cells may be diploid, similar to the mother cell, or may be haploid. The genetic materials becomes half of the mother cells, right? So this is the definition of the cell cycle. Another point, for the cell cycle, it takes minimum of 12 hours. What is the time for the cell cycle? Minimum. This 12 hours is minimum. Understood? So the minimum of the 12 hours it takes. How much it takes maximum? Maximum it may takes years. We will discuss in the meiosis how it takes years. In the oogenesis, right? During the oogenesis, some of the cells that takes years and years, up to 50 years for the completion of the meiosis 2. So that minimum duration for the cell cycle is how much? 12 hours, and maximum time may be years and years. So you can memorize like this. Now, what are the stages of the cell cycles? The cell cycle stages we can classify as go, Sally, go, make children, right? So what are those? Go, Sally, go, make children means go, Sally, go, make children, right? Go for what? G1 phase. Sally, S phase. We have done this already. This is just the revision. Another go, G2 phase. Make, maybe mitosis or meiosis. Children, that is cytokinesis. Let's see. Above this is called what? Interphase. And this mitosis or meiosis, they will further divide into what? P, M, A, T, right? That is what? Prophase, metaphase, anaphase, and telophase. Now, one point very important you have to understand over here. See, one mitotic division, right? To complete one mitotic division, it has only one interphase. Done. So in case of meiosis, one interphase followed by two meiosis, meiosis 1 and meiosis 2. Hope you understand. One interphase followed by one M phase in the mitosis, one interphase followed by two M phase in the meiosis. Hope you understand, right? So that finally in the meiosis it produces the haploid daughter cells, right? So you understand this one. So these are the stages. It's the same thing we have just now we have discussed, right? So we have the G phase, S phase, G1 phase, S phase, G2 phase, and we have the M phase. And sometimes in the G1 phase the cell may enter into the G0 phase. If it enter into the G0 phase, the G1 phase becomes longer. If it does not enter into the G0 phase, the G1 phase becomes shorter, right? So do you understand what I said over here? Note, during the G1 phase, if cell enters to G0 phase, the G1 phase becomes longer. If it does not enter into the G0 phase, the G1 phase becomes shorter. Hope you understand, right? Yes. Now, let's discuss a little bit here about the timing of the cell cycle. So minimum timing of the cell cycle, what we say, that is about 12 hours, right? So let's discuss here. This is my which phase? G1 phase, and this is my what? The S phase, and this is my what? G2, and this is my what? M phase. Yeah. So what is the timing for this? Usually for the G1 phase, G1 phase we will discuss after 1 minute. Let's go to the S phase. For the S phase, it takes about 6 to 8 hours. How much? 6 to 8 hours. For the G2 phase, it takes about 2 to 4 hours. 2 to 4 hours. And for the M phase, it takes about 1 to 2 hours. Why we didn't discuss the G1 phase over here? Let's see. This G1 phase, it has the R restriction point in the middle. Which point? R point, restriction point. If the cell do not cross this restriction point, the G1 phase becomes long. If the cell crosses the R, the restriction point, after the completion of the G1, they will continuously go to S, they will go to G2, and then they complete the M phase. That means what is here, we can check the cell cycle initially, that is the R point, restriction point. That's why G1 phase is variable. If the cell is rapidly dividing, if the cell is rapidly dividing, in this case, the G1 phase is short. In this case, the G1 phase is usually of less than 2 hours. What defines the length of the G1 phase? That is what? R point, the restriction point. If it crosses the restriction point, now there is no restriction to the cell cycle. It will continuously go to the S phase, G2 phase, and M phase if there is no DNA damage or DNA mismatch, right? If the no DNA mutation occurs after crossing the R checkpoint, the cell cycle does not stop, it continues. That's why S phase is specific 6 to 8 hours, G2 phase is specific 2 to 4 hours, M phase is also specific 1 to 2 hours with some exceptions. That exceptions we will discuss. But G1 phase is variable. Why it is variable? Because it has the restriction R point. If there is the growth factor, then only it can cross the R point, restriction point. If it crosses the R point, then the cell cycle continues. Hope you understand. So what do we need to cross the R point? We need mitogens. What do we need? Mitogens means those factors which stimulates to cross the R point and the cell undergoes the cell cycle smoothly. That is what mitogens. So the details of the mitogens, the mechanisms, the pathway we discussed already in the different, the phases of the cell cycles. After this, let's see. So the minimum time required for the G1 phase where the cell is rapidly dividing is how much? Less than 2 hours. So sometimes it may takes more than 100 hours in the same cells. So minimum it is how much? Less than 2 hours. Maximum it may be more than 100 hours. If there is the deficiency of the mitogens, or if the cell enters into the G0 phase and it has to exit through the G0 phase and then go to the S phase, then the time becomes longer. This is one variation. Let's talk the another variation over here. The another variation is what? Let's see. The primary oocytes in the gametogenesis. Primary oocytes. This variation we are discussing for the M phase. The primary oocytes in the the M phase. These primary oocytes, they forms during the 5th month of IUL, and these primary oocytes in the 5th month of IUL, they go and they stop within the diplotin stage. Which stage? Diplotin stage of M1, means meiosis 1 of prophase 1 stage. Which one? Prophase 1 of meiosis 1. The primary oocyte stuck in which stage? Diplotin stage. In which month? 5th month of IUL. And they complete this during the puberty. It may, they complete this during the puberty. During the puberty means what? We have multiple primary oocytes which are stuck within the ovary during the 5th month of IUL. Now, when the menstruation cycle begins by the puberty, every month 1 or 2 primary oocytes only mature. So that those primary oocytes who begins to mature during the the respective menstrual cycle, those who stuck in diplotin, they will complete their M1 phase, prophase, and then they enters into the M2 phase, right? But those primary oocytes which because one menstrual cycle only 1 or 2 primary oocytes develop. So the female, they have the menstrual cycle which begins from the puberty, it goes up to the 45 to 50 years. So turn wise, this month one completes the meiosis, next month the another will complete the meiosis, the next month another will complete. So likewise they wait for their turn from the 5th month of IUL till the 50 years of age, so that the M phase, the M phase usually is of 1 to 2 hours, but the variations we discussed here in the meiosis, right? So the primary oocyte which stocks in the diplotin stage of the prophase 1 of meiosis 1 during the 5th month of the IUL, they complete their cycle maybe by the age of 50 years. That's why this is one variation. Another variation here, these primary oocytes, when they becomes a secondary oocyte, these secondary oocyte, they further go division and they get stuck in the metaphase 2. Metaphase 2. These metaphase 2, they will complete only when the fertilization occur. Otherwise, without fertilization, they will involute, they will die. So that this fertilization, after the ovulation till the fertilization, the time may be, the time taken may be 2 to 4 days, right? 2 to 4 days means what? The viability of the sperm. They will wait until the sperm come and the fertilization. If it cannot fertilize, this secondary oocyte also degenerates, right? So that this secondary oocyte, they complete the M2 phase, that means meiosis 2 phase, if the fertilization occurs. And the duration for the fertilization is how much? 2 to 4 days. That means what? They will wait until the sperm comes and fertilize. If the intercourse does not occur during that period, the fertilization does not occur. If the fertilization does not occur, they do not complete their meiosis 2. That means the M phase also has some kind of variations, right? So we discussed this one. So let's go to the next point here. This is cell cycle, just now we discussed, right? So in this cell cycle is driven by the cyclins proteins, plus cyclin dependent kinase proteins. Which proteins? Cyclin proteins and cyclin dependent kinase proteins we need to drive the cell cycle. Till now we have more than 15 types of cyclin proteins discovered, right? How many cyclin proteins? About 15 or more than 15 types of cyclin proteins has been discovered till now. And the cyclin proteins, we give the name as what? Maybe the cyclin A, cyclin B, cyclin C, cyclin D, cyclin E. We give the name accordingly, right? So these are about more than 15 types of cyclin proteins discovered. And CDKs are the drivers of the cyclins. So they too combinedly, they carry out the cell cycles. We will discuss about the cyclins and CDKs in short. And the cell cycle is driven by the dream transcription factors. Dream transcription factors. This dream transcription factors, we consider these as the controller of cell cycle. And how is it the controller of the cell cycle? That have been discussed already in the earlier videos. So you watch about the dream proteins of the dream transcription factors. Okay, if there is dream activation, then the cell do not become age. The aging of the cell will be delayed if the dream activates. That's why dream is also called as the anti-aging protein or anti-aging transcription factor. So this is nowadays is being used in the the genetic research, like to delay or prevent the aging. This is a very important protein. In the future, it may gives the very good milestones in the genetics. So let's discuss here about the CDKs, right? So that a little bit, it's just a review, because we have discussed this a lot already in the previous videos. This is my what? G1, this is my S, and this is my what? G2, and this is my M phase, right? So this CDK proteins, they appears in the sequence of D, E, A, B, right? So what are the sequence? D, E, A, B sequence. These are the cyclins. These are what? Cyclins. And CDKs, we have the CDKs, they appears in the sequence of 42, 21. If you remember, we discussed these previous lectures, right? So 42, 21, these are what? CDKs. So what happens here? That means what? Cyclin D and CDK4. Cyclin D and CDK4, it appears the first. This is a question in the exam. Which cyclin protein rises first during the cell cycle? That means cyclin D protein rises first during the cell cycle. Which CDK protein rises first during the cell cycle? That is CDK4 rises first during the cell cycle. These are the questions, please remember, very very important questions. So cyclin D and CDK4, they appears at first. They appears which stage? In the G1 phase to cross the R checkpoint. So once they cross the R checkpoint, the second set of cyclin, they appears, that is what? Cyclin E with CDK2. That is my second set, cyclin E with CDK2. So that the cyclin D plus CDK4 comes in which stage? Phase? G1 to cross the R checkpoint. Hope you understand. Now the second is what? E and CDK2. This E and CDK2, they appears during the S checkpoint. Which checkpoint? Sorry, G1 S checkpoint, not the S checkpoint. What is this one? G1 S, to cross the G1 and reach to the S. They appears during which stage? G1 S checkpoint. After that, the next set will come here. What is that next? The next set. The next set is cyclin A with CDK2. Cyclin A with CDK2 will appear here to cross the S checkpoint. Which checkpoint? S checkpoint. This is my what? The cyclin A with CDK2. This cyclin A and CDK2 will follow whole this one. Cyclin A and CDK2. Finally, it reaches to this checkpoint. This checkpoint is what? G2M checkpoint. What is this one? G2M checkpoint. So what left here? R checkpoint, S checkpoint, R checkpoint. This one is G1 S checkpoint. This one is G1 S checkpoint. Finally, it goes to where? This one is where? This one is S checkpoint. So finally, we reach to B and 1. Cyclin B with 1. This cyclin B with 1 will come in where? G2M checkpoint. Which checkpoint? G2M checkpoint. So this is my G2M checkpoint. So you will just follow the sequence as what? D, E, A, B. That means D appears in the R checkpoint. D and CDK4, right? So R checkpoint, D with CDK4. G1 S checkpoint, E with CDK2. S checkpoint, we have A with CDK2. And we have what? This will drive whole to here up to G2M. And in the G2M checkpoint, what comes? Cyclin B with CDK1. So hope you understand this is the sequence, right? So we have discussed this already a lot. And we go into the types of cell divisions. Our topic was what? Mitosis and meiosis. Actually, the mitosis is what? Equational cell division. Equational cell division. So that it maintains the genetic stability. Maintains what? Genetic stability. That means the mother cell is equal to the daughter cell, right? So that they maintain the genetic stability. But the meiosis is what? Reductional cell division. That means what? Mother cell is daughter cell here, but half of the mother cell only will be equal to the daughter cell, right? So because the genetic matrix becomes half. So let's discuss a little about the mitosis and we go to the phases. So from the mitosis, what happens? This is a mother cell. it will divide into the two daughter cells with the same genetic materials, right? So it is what? Number one, we can say it is equational. It is equational. Number two, it produces genetically identical, similar two daughter cells, right? Another point, this occurs in the somatic cells, and it is useful in wound healing. It is useful in tissue repair. If you have the injuries, we need what? Mitosis. It is important in the tissue growth. Hope you understand, right? So the chromosome numbers remain what? See, we have here M, that means mother, we said, yes or no? Daughter also has the M and M, that means the chromosome numbers remain the same. This is for the mitosis. So look at here, the mitosis takes approximately 1 hour. We discussed already M phase 1 to 2 hours, right? So 1 to 2 hours, that means we can say here approximately 1 hour to 2 hours to complete the mitosis. And mitosis completes in how many stages? Further 4 stages. What are those? People meet and talk, PMAT, right? So P for what? Prophase, M for metaphase, what is this one? A for anaphase, and another one T for what? Telophase. Prophase is earliest and longest. Prophase is what? Earliest and longest. Anaphase is the shortest phase. Right? So prophase only takes about 30 minutes. Remaining others takes about 30 minutes. So that the mitosis, it completes in how many stages? 4 stages. That is what? Prophase, metaphase, anaphase, and telophase. That is people meet and talk. That is PMAT. And you can see here mitosis as mother cell forms two daughter cells which are diploid and genetically they are identical, right? So they involve in the growth and repair. Meiosis we will discuss in the next class. So let's see here about the prophase. Prophase is what? The longest phase, and I already mentioned that it takes about how long? About 30 minutes to complete. And the chromatin begins to condense, and the centrosome splits into two and moves to the two opposite poles, splits into two and they moves into two opposite poles, centrosomes. The mitotic apparatus, what is this mitotic apparatus? See, we have the chromosomes in the middle, right? These chromatins, these chromatins, they begin to condense and they begins to become thick. At the same time, we have what? Centrosome. This centrosome split and then moves into two opposite poles. As they moves into two opposite poles, they begins to generate the microtubules. These microtubules are called ester formation. What are they called? Ester formation. So these ester formation microtubules, these esters, they begin to arrange in the MTOC, microtubule organizing center. In the microtubule organizing center, how do they organize? They will organize in what? If you remember, we have the two, the microtubules here, and we have the three, the triplets will be here. Triplets. 9 group of the triplets will be in the periphery. So that, how do we call this one? 9 triplets plus 2 at the center, right? So this arrangement occurs. This arrangement is called what? MTOC, microtubule organizing center. This arrangement will begin to form what? The spindle fibers. Spindle fibers. Understood? So this ester formation with the spindle fiber organization, this combined is called mitotic apparatus. What is mitotic apparatus? That is ester formation plus what? The spindle fiber formation. This combined is called what? The mitotic apparatus. At the end, what happens? All the Golgi body, endoplasmic reticulum, and the nuclear membrane will disappear. Will resolve. Is that clear? So what are the events occurs in the prophase? Prophase is the longest phase. Chromatin begins to condense and they become thick. The visible. the centrosomes divides into two and then moves to two opposite poles. By reaching the opposite poles, they begin to generate the esters. Esters means microtubules. These microtubules undergoes MTOC, microtubule organizing center, and then they becomes 9 multiplied by 3 plus 2. That means what? These 9 triplets at the periphery and then the 2, the duplets at the center. So that this arrangements will form the spindle fibers. So these esters with the formation of the spindle fibers combinedly is called what? Mitotic apparatus. So this is a question in the exam. Mitotic apparatus will appear in which phase of the mitotic division? This is the prophase, right? Then what happens at the end? The Golgi body, the endoplasmic reticulums, and the nuclear membranes will degenerate. Now we'll go to the metaphase. What is the metaphase? In the metaphase, the chromatins condensed, and the chromosomes become shortest you can say, and thickest. So that this is the most accurate phase for the chromosomal study. That is also called what? The karyotyping. This is a question in the exam. So in which phase of the mitotic division or the cell division it is perfect for the study of the chromosome? That is what? The metaphase stage, right? So during the metaphase, the chromatin condensed further and they become shortest and then thickest, so that they are clearly visible. So this is the perfect stage for the study of the chromosomes. That is what? Karyotyping. So they can ask you in the exam, karyotyping is perfectly done in which stage? That is metaphase stage. After that, what happens? The what is this one? The center portion of the chromosome, that is my centromere. This centromere that begins to express the sticky protein over here. These sticky proteins which appears within the begin to appears within the centromere, they are called what? Kinetochore formation. What is this? Kinetochore formation. So that what happens? The spindle fibers which were forming from the what is this one? Centrosomes. These spindle fibers, they begin to attach with the kinetochores. And then they push the chromosomes to the central of the cell. You can see in the picture, center of the cell, so that all the chromosomes, they arrange in the equatorial plane. Central alignment of the chromosome. What happens? Central alignment of the chromosome occurs. What is this one? In the equatorial planes. Kinetochore formation occurs. Hope you understand, right? Kinetochore formation occurs, and then the attachment of the spindle fibers with the kinetochores occurs, and then they push the chromosomes to the center from both the sides. So the chromosomes, they will arrange in the central line, right? This is what? In the equatorial plane. Hope you understand. Yes, this is also called what? Metaphyseal plate or equatorial plate. What is this called? Metaphyseal plate or equatorial plate. This plate. Hope you understand. This is the metaphase. So metaphase, what are the stages? The chromosomes, the chromatids, they condensed and they become shortest and they become thickest. So that this is the best stage for the chromosomal study for the karyotyping. First point. The second point, what? What is this one? The centromere, they begin to express the kinetochores. That means the attachment site for the spindle fibers. So the spindle fibers, they begin to attach with the kinetochores in the centromere, and then they push from both sides the chromosomes at the central equatorial plane. That is called what? Equatorial plate formation or the metaphyseal plate formation. These are the events during the metaphase. Now let's go to the, you can see here, these are the microtubules. Microtubules has what? Triplet, 9 triplets, and we have what? 9 plus 2 arrangement, and you can see here, these are the spindle fibers. These spindle fibers, they attach and then they are pushing these chromosomes to the center, to the equatorial plane, right? Now let's go to the anaphase. Anaphase we discussed already. This is the shortest phase. So what happens during the anaphase? These spindle fibers from the both sides, these spindle fibers, they begin to shorten. And this is my chromosome, this chromosome has two sister chromatids. During the S phase, the DNA was replicated. During the S phase, which phase? S phase, the DNA was replicated, and then there were two DNAs in each chromosome, right? So that these sister chromatids are what? Two separate DNAs. So during this anaphase stage, my spindle fibers, they begin to shorten. Spindle begins to shorten. If the spindle fibers they begin to shorten, they will pull this chromatid to this side, and then they will pull this chromatid to the opposite side. So here is my what? Centromere. This centromere has which protein? Cohesin protein, as we discussed earlier in the question, right? So this cohesin protein now begins to separate, so that my centromere will split into two parts. So that what happens? Half of this will move upward, half of this will move downward. That means two opposite poles. So that what happens? Two sister chromatids, they begin to move to the opposite pole. Sister chromatids begin to move to opposite pole. During which phase? Anaphase stage. This is the question they will ask you in the exam, so please remember this one. This is the stages, right? So so they move to the two opposite poles. Let's see here. This is the picture we have. So during the interphase, we have all the events that we discussed already, and during the prophase, what happens? We have the two what is this one? The centromeres, they are moving to opposite poles, and they begin to appear what? The esters. And after the ester formation, or the spindles, they begin to form, and once the spindle is formed, we call this as what? The mitotic apparatus they form. And what happens during the metaphase stage? The chromosomes, they align centrally in the equatorial plane, so we call this as what? Metaphyseal plate or equatorial plate. And then during the anaphase, they begins to pull to opposite side, and see here they begins to form two nucleus and two sides. And see in the telophase, that what happens during the interphase, the nuclear membrane was disappeared not in the interphase, prophase, the nuclear membrane were disappeared. Now the nuclear membrane reappear. The Golgi apparatus and other the cell organelles will disappear. Now they begin to reappear here. Yes, so this is about what? Telophase. So let's go to the telophase here. So what happens to the telophase? In the telophase, see, during the prophase, what happens? The chromatin begins to condense, but in the telophase, it is opposite of the prophase. So the chromosomes begins to become chromatins. That means the proteins which were forming for the chromosomes, the coiling occurs. So those proteins, they begin to dissolve. So that the chromosomes, they begin to change into what? Chromatins, right? So they decondense. Decondensation occurs. What occurs here? Decondensation of the chromosomes. Condensation of the chromosomes occurs, so that they will become again the chromatins. So see, in the earlier phase, what was here? Individual chromosome could be studied, but now here they all become like this coil structure. So the individuality will be lost. Individuality, that means we cannot study the chromosomes individually now. So individuality of the chromosomes will be lost, because all of them becomes the chromatins, right? So nuclear membrane begins to appear. Similarly, the Golgi body, the endoplasmic reticulums, all the cell organelles also begins to appear. So finally they becomes two distinct daughter nuclei in the two pole of the cells, right? So after that, what happens? The cytokinesis undergoes. During the cytokinesis, what happens? The cell membrane begins to divide by the furrow formation. What is this one? FURROW, furrow formation, the cleavage formation in the animals, and the cell plate formation in the plant. Like this, so that they undergoes what? Cytokinesis, and finally they forms the two daughter cells. Finally they form what? Two daughter cells which are identical to each other, which has the the same genetic material as like the maternal cells or the mother cells. That is called what? Diploid. What is this one? Diploid means two sets of chromosomes are present. That means one paternal set, one maternal set of the chromosomes are present. Hope you understand the mitosis. Tomorrow we'll discuss about the meiosis. So till then, thank you very much. Hope you enjoyed the lecture.